Okay, it's a great pleasure today to have Hyunseok here as our first speaker in 2026. Hyunseok, today it will talk about topological flowscape, revealing state transitions in non-reciprocal living matter. He is an experimental physicist specializing in active matter and microbial ecology. He is an independent postdoctoral researcher in the Department of Physics at the MIT, where he investigates dynamic states in non-reciprocal starfish embryo systems and in microbial communities, using a combination of modeling and experiments. He received his PhD in physics, also from MIT, under the supervision of Jeff Gong, studying emergent behaviors in microbial communities. Okay, so we are glad Hyunseok is here, and now it's your time. You can start now.
Sure. And can you hear me well? Yes. Okay, and thank you very much for the introduction and the invitation. I'm very excited to be the kickoff speaker for SLAM seminar this semester. And in this talk, while indeed I worked in microbiome ecology as well, today's talk will be mostly about starfish. In particular, I want to tell you a story about how we map a living matter of starfish on the left to a landscape on the center and eventually to a flowscape on the right. In particular, I aim to share why we had to do this and what we gained through the process. And of course, this work was possible only because of the amazing teamwork from three different labs and these funding sources, which I greatly appreciate. And today, I want to start with a pretty broad notion that in many of soft, living, active, adaptive matters, intricate forms emerge from collections of these units, tissues from cells, biofilms from microbes, and also flux from birds. Importantly, these forms are not programmed top-down, but are self-organized through interactions between these entities. These self-organized structures can be far richer than the non-living counterparts, as a wider possibility of interactions are allowed. In particular, for this project, we are interested in a type of interaction which is called non-reciprocal. Newton's third law states that action and reaction is equal and opposite, which is always true at microscopic scales. But in fact, in equilibrium, the action-reaction symmetry holds even for effective interactions mediated through the environment. Nevertheless, living systems are fundamentally non-equilibrium, and their action and reaction now no longer need to be symmetric, like how a hare will, let's say, pull the wolf to be closer, while the wolf will push the hare to run away. So these non-reciprocal interactions are particularly relevant for living systems, but how they affect the self-organized structures still remain elusive. Our lab uses experimental system of interacting embryos to study how living systems can self-organize structures emerging from interactions, like one in the video I'll show here. We are looking at a top view of a small well filled with seawater. The system spans few millimeters, which is actually much larger than the individual starfish embryos shown here. And over hours, we'll see that more embryos appear in this field of view and self-organize interesting structures, which I will play now. As we can see, the embryos form a remarkably ordered crystalline structure that also self-sustains a chiral dynamics, which is unfortunately maybe not pausing, but it will start to basically rotate in a clockwise direction as a whole crystal. And this is why we call this system a living chiral crystal. And the question here is, what kind of interactions underlie this self-organized structure, and how will we change this? On the left, we see a side view of a single starfish embryo zoomed in, 24 hours after fertilization. In nature, this embryo would need to survive in the ocean, and it turns out to be a good swimmer. Makes sense? It makes strokes with its cilia, which generates hydrodynamic flow so that the embryo pushes the fluid downward and arises to air-water interface. Now from the top view, we see that these embryos are chiral, rotating clockwise due to the chiral symmetry breaking in its ciliary activity. Moreover, we can see that this embryo sucks the fluid toward itself on the plane as it pushes the fluid downward. This inward flow leads to an effective long-range attraction between a pair of embryos so that they form a stable bound pair. In addition, the chiral rotation of each embryo makes the pair rotate as well. And these pairwise interactions led to the self-organized crystal we saw in the previous slide. So how did this platform allow us to study non-reciprocity? Building up on the same embryo pair, we tune their interaction by introducing developmental time gap. The embryo on the left, E1, is the one we used previously, and the embryo on the right, E2, is older than E1, so it's larger, more elongated, and generates a quantitatively different hydrodynamic flow from what E1 generates. As a result, as we can see in this top view video, the embryo pair exhibits a striking run and chase dynamics. And this tells that E1 are pushing E2 away, while E2 is pulling E1 to follow. This run and chase dynamics, mediated by the hydrodynamic flow in the media, highlights that the non-reciprocity between E1 and E2 embryos. Now, let's see how this non-reciprocal E1 and E2 pair realizes a different self-organized structure. Again, we see a top view of a well plate filled with seawaters and thousands of starfish embryos ready to come up to air-water interface. Since now we have two different types of embryos, we mark E1 as blue and E2 as red, overlaying their 2-minute trajectories on the video. Let's first see the 3 hours of dynamics. I hope the video sees well. Is the video playing well? Okay, great. Yes. Instead of forming a single crystal, these embryos are forming multiple clusters. And each individual cluster is now quite dynamic, with its E2 region leading E1 region, like in the pair case. Also, while these clusters can transiently bound to form a larger one, the non-reciprocal interaction destabilizes those and break them apart, sustaining their dynamics for hours.
Here's a quick clarification.
Yes,
Here it's a 50-50 mixture of
that's a
events.
great question. We tried to make it 50-50 mixture, but this basically what happens is that we make the initial mixture to be 50-50,
Ah,
but we do not necessarily control how many of the embryos that come up to the interface.
oh yes.
And indeed, this is a great point because if you are following it more closely, we can see that compared to the initial maybe like 30 minutes, the later, let's say the composition of the whole system has slightly different composition. However, what we found was that after the initial 30 minutes, the ratio between the E1 and E2 stays relatively fixed over the time of the experiment that we are focusing on.
Okay, thank you.
Great. Thank you for the great question. Yes.
Is
And
there a size difference in terms of radius between the E1 and the E2?
size in terms difference in terms of the radius of the E1 and E2, of course. So that's maybe most visible when we go slightly before to this slide.
Yes.
Yes. So in this slide, basically what we find is that the size difference exists between E1 and E2. Roughly, if you quantify it, we will show later that what we do is also to numerically infer the infer the interactions between the embryos, and that turns that these two embryos are roughly maybe like 10% difference in terms of
Hmm.
their effective size of the. So one is let's say 20 micrometer scale, and the other is more like 220. Sorry,
Okay.
200 versus 220. All right, yes. And please interrupt me for more questions. I will have some time for addressing this. So yes, this was what happened in the first 3 hours. But it was not the end of the story, because after hours of dynamics, for example, in this video, which was like 5 hours from the onset of the experiment, the self-organized structure transitions to a single cluster, and now the embryos are instead of traveling far away, just jiggles around near their lattice point. And to quantify the system level transition over time, we first tried a measure of dynamic order, which we call velocity polarization. This velocity polarization, P, is basically a system average of unit vectors pointing in each particle's velocity direction. In other words, it is a measure of how the embryos are moving together in a line direction if the magnitude is high, or not if magnitude is low. Now, let's see how this polarization changes over time. For the initial 3 hours, we find that the velocity polarization can reach high magnitudes, while its direction can change, and it clearly shows that the embryos are traveling together. However, later on, the velocity polarization dies out, and we see that the embryos no longer travel, with their positions simply fluctuating in random directions. And what this plot highlights is the abruptness of the transition. Instead of continuously relaxing, these embryos stay in a traveling state for hours, then transitions to a fluctuating state, staying there for another hours. And this was quite confusing for us. Why 3 hours? What happened at the time? We realized that these different interactions, behaviors can happen because our non-reciprocal interactions can also change over time as embryos develop during the experiment. So by using a recently developed interaction inference techniques from our collaborators, we quantified the effective pairwise interactions between the embryos. From the inference on the early time, where E1 is 25 hours old, we find that the longitudinal interactions consist of the long-range attraction and short-range repulsion, and basically the equilibrium distance of these two interactions, one going from E1 to E2 and the other going from E2 to E1, are quite different. And because of the difference, the embryo pairs will exhibit non-reciprocity in the distance regime where E1 is repelling E2, while E2 is attracting E1. And now, as we do this inference based on embryo trajectories over a later time window, we can see that the development over our experiment makes difference, bringing the E1 embryos to be more similar to the older E2 embryos as it ages. And this time dependence is quite nice, in fact, because it allows us to study not just the self-organized structure, but the whole self-organization process driven by the transitions in the interactions between these individuals. So far, we have observed
Hyunseok, sorry, another quick clarification.
Yeah.
Is there a way to understand this transition in the interactions from just looking at the sizes and the change in the hydrodynamics, which will the strength of the sort of Stokes, the rotelet or Stokeslet that you guys model, if I remember.
Yes, that's a very good point.
Members, is there a way to
Yes.
please rationalize?
The short answer is that we will need a little bit more complicated model.
Okay.
One very nice way to show this is to go back to this side view video of the two embryos. What happens on the side view is that the embryos are keeping almost dancing with their axis during the procession. And the key point is that the procession is a little bit asymmetric, such that this E2 embryo in particular will tilt away further in terms of angle when it's tilting away from E1 compared to the other direction where it's tilting toward E1. And what we found is that this kind of tilting from the top view indeed synchronizes with the degree of the non-reciprocity, like the run and chase dynamics.
I see.
And we think this is definitely at least one of the mechanisms that's driving. And we thought that it is maybe like quite different to recapture with the minimal model. And that's why one of the reasons why we were actually going into this more data-driven inference to allow the mechanical insight.
Sounds good. Thank you.
Right. Thank you. Yes. Okay, so we have observed a transition in non-reciprocal starfish embryo mixtures from multiple clusters traveling together to a single crystal fluctuating. And we also found that there is an underlying change in the interaction between embryos that seem to decrease in its non-reciprocity. So it seems that we have a mapping between these two non-reciprocal interactions and the two corresponding states. But up to this point, it's quite empirical. And what I'll tell you now is how we made this mapping more rigorous. Basically, instead of relying on embryonic development to change interactions, we turn to numerical models where we can systematically tune interactions by ourselves, where the pairwise interactions just stay the same as inferred, except these longitudinal forces. The key parameter is n, which is non-reciprocity of this pairwise interaction, that scales the difference between the two interactions, so that when n is 1, the interaction is exactly the ones inferred from the experiment. When n is 0, the interaction is perfectly non-reciprocal, being the average between the two. And when n is greater than 1, the interactions are more strongly non-reciprocal than in the experiment. And as we tune non-reciprocity in our model, we observe these many states of non-reciprocal mixtures. And now I'll walk through some important examples. First, for some small non-reciprocity n smaller than 1, we essentially observe stable crystals. As non-reciprocity increases, maybe these stable crystals might be more like self-propelling crystals, which has non-zero polarization due to non-reciprocity. But nevertheless, in terms of self-organized structure, it will remain crystalline. A second example is a case where with experimentally inferred interaction n equal to 1. And in this case, we see a quite intricate dynamics where clusters repeatedly break and merge. And I want to emphasize that this dynamics persists for a very long time in the simulation, just as we already saw in our experiment early state. And finally, for strong non-reciprocity, now we basically see some fragment instead of a more repeated cluster merging and breaking. And this, with this, we are almost there for a bottom-up mapping from non-reciprocal interactions to emergent self-organized states. And for the interaction side, it is quite simple. Now, since we have a non-reciprocal parameter n, it provides a sense of quantitative comparison between the interactions. However, there's a challenge in terms of the resulting self-organized forms. Now, with all these dynamic and complicated forms, it is not obvious how can we even make a quantitative comparisons. And this is where topology enters our story. What inspired us is the concept of topological transitions, especially the ones called T1 transitions, which has been already used in self-organized structures such as tissue dynamics and morphogenesis. So let's just quickly go through it. Let's imagine, like in panel A, we are zooming into a monolayer of cells A, B, C, and D, and C and D are the nearest neighbors. Here, each cell's positions, maybe in nearity, will fluctuate slightly, but such small fluctuations will not make radical changes, and each cell will keep its neighbor. But then, if you inject sufficient energy, then this may deform the system so it can pass through the middle configuration and settle down to a new configuration where A and B are now the nearest neighbors. If you make this notion abstract, it is basically a transition in the graph of nearest neighbor network, marked red here in terms of the edges that are transitioning. And this graph can be a network of cells, but also basically a network of anything like starfish embryos. So we also map our embryos into such neighbor connection network. In particular, for each embryo, we characterize its next to nearest neighborhood as a topological motif. For the embryo in the bottom right, for example, its local neighborhood corresponds to a perfect crystal with all nearest neighbors having 6 neighbors. On the other hand, for another embryo, its neighborhood does not seem that symmetric. Now, the whole spatial structure that we see in a snapshot of the video becomes a distribution of topological motifs. But at this point, the distribution itself doesn't give much intuition of what the key features of our system is. And we need another step to better understand this distribution. What we notice is that these motifs have a physical sense of pairwise distance in terms of the number of T1 transitions between them. This topological distance, which is the minimum number of T1 transition we need to convert one motif to another, provides a sense of space where all these 15,000 motifs we see observed from experiment and simulations do live on together. And at this point, we thought it would be great if we can, instead of assigning just random index to each motif, we capsulate this high dimensional space where motifs actually live and see which regions of motifs are the most important. And the trick we used here is to use dimensional reduction. For example, in our case, by using what's called MDS or multidimensional scaling, we found a two-dimensional manifold where the pairwise distance between these motifs are well preserved. In other words, we assign coordinates to each motif so that motifs who are topologically close together will be put close together in terms of the low-dimensional manifold. And finally, what we use is the frequency of motifs as the weight in this embedded space, thus converting the structures into a landscape of local network topologies. In particular, like in these topographic maps, the landscape can have peaks of most probable configurations and valleys of least probable configurations. So now let's see what this map looked like. For our simulation with small non-reciprocity, which resulted in a stable crystal, we get a landscape, topological landscape with a single peak. Now, as we increase non-reciprocity, we find something very interesting. The single peak landscape now has become a double peak landscape with a new peak appearing somewhere in the slightly on the right. And finally, for a stronger non-reciprocity case, which resulted in a fragmented structure, we see that the strong peaks has disappeared. And to better understand this, especially the transition between the low non-reciprocity and the infinite non-reciprocity, we now go back to the original topological motifs and mark the most dominant 14 motifs on this manifold. And what we find is that these peaks correspond to the two most dominant motifs. First, the peak on the upper right corresponds to a perfect crystal, which we call M1. And another peak on the lower left corresponds to an almost crystal with a 5 defect, which we call M2. Now, to get a quantitative view, we slice through this M1, M2 axis and see how non-reciprocity drives transitions between these structures. Here, what we are plotting is
Can I ask a quick question about how is this a continuous space if it's about neighbor connections? What fills in the space in between one and two?
That's a very good point. So we use basically a standard rule of thumb for a small smoothing. So the first thing that we might need to remember is that while I'm showing only 14 points, in principle there will be like 1,000 times more points in this. And nevertheless, as you said, this will still be a discrete number of points. And what we are showing is basically a smoothed out version using some standard Gaussian kernel with the standard rule of thumb for the width of the Gaussians.
Okay.
And we think that, yeah, philosophically, it is trying to find the region of the most likely motif. And in principle, while our example shows that one peak corresponds to one motif, we can easily imagine a case where one peak actually corresponds to a multiple, let's say, collection of motifs. And in such cases, this kind of treatment can also be able to be useful in terms of finding such collection of the important features.
But so as we walk along this line, is there a specific like movie you could play that connects the red squared and the blue squared configurations?
Mm, could you clarify the connection in terms of the maybe like dynamics of transition or?
Exactly. Is your red line that you've drawn describing a transition that that one could animate, for example?
Ah, I see. Yes, one could do that.
Okay,
Yeah.
that's
Yes.
good for now. I'll let you continue.
Yes, but I
Thanks.
will do, yes, always more. So basically, yes, before we jump in, maybe what I want to clarify is that in the slide here, what I'm showing in the center is the topological landscape for a single non-reciprocity. And there will be each landscape for each non-reciprocity values that we tried. And what we want to visualize is the trajectory of how this landscape transitions along the changing non-reciprocity. And that's how we arrive at this slice with now each landscape being visualized as a curve on this M1, M2 slice. And this curve, I'm doing a small modification so that the y-axis now is a negative log likelihood, which can be intuitively thought as some energy landscape, as it means that the system likes to be in the bottom of the well. So this is kind of crude intuition. And now I'll walk through the curves. When non-reciprocity is 0, we can see that M1 is the most likely configuration in this curve. And when non-reciprocity is 0.75, M1 is still the most likely configuration. However, as we increase non-reciprocity further to n equal 1, we now see that M1 and M2 are equally likely. In other words, the M1 and M2 are coexisting. And finally, as the non-reciprocity becomes even greater than 1, we see that M2 now is the more likely state than more likely configuration than M1. And this analysis is very useful for us in two ways. First, it clearly shows that the non-reciprocity essentially transitions the system from a M1 dominated state to M2 dominated state. And second, the way it transitions, which is through a coexistent state, brings the analogy of something like water turning to ice, or technically a first order phase transition. And before we go deeper, let's go back to our experiment. And this will be now a video because like what we did for simulations, we could also construct topological landscapes for experiment and see how it changes over time instead of changes over non-reciprocity.
Sorry.
Yes.
I have a question. Apologies,
Sure.
you already mentioned this. So, your topological motifs, how do you decide how many points to use?
That is a great point. We decided to use two, which is in some sense the smallest number of smallest scale of the neighborhood where things start to be non-trivial. Because if you choose neighborhood of size 1, that will essentially turn to neighborhood size, basically the number of neighbors, which will lead to a one-dimensional, let's say, space in terms of the topology. Um, the philosophy for doing this, okay, there are two types of reasons why we choose n equal to the next to nearest neighborhood. One practical
wait a
reason.
second. When you draw these diagrams, they seem
Yes.
like you're only looking at next nearest neighbors.
Yes.
Oh, it looked like a lot of points to me. Oh, okay.
Or in other words, if I go back. So indeed, there will be a lot of points in terms of the whole network topology, but what we do is that we just count the distinct next to nearest neighborhood, like one we highlight here. So that, for example, if we had a crystal, even if the crystal has 1,000 embryos, if it was a perfect crystal, all the motifs, topological motifs that are in terms of the local neighborhood of each embryo will correspond to a single perfect crystal next to nearest topology,
Yeah,
which you
I was
call
just saying that. I was just saying that when you showed the motifs, it looked like each motif had like, I don't know, 20 points or something like that. And those are all just next nearest neighbors.
Yeah, they are all the next nearest neighbors, yes.
Okay.
Okay. All right. Any other questions before I move back to the experiment?
I actually had another question. So you saw this abrupt change from the I forgot what you called it, but in your P, there was like an abrupt change at some number of hours. Is
Yes.
there and similar, is the effective interaction also abruptly changing, or is it gradually changing and then you have some kind of phase transition?
That is a excellent point. I don't think we have enough, let's say, statistics or the statistical resolution to call that it's a abrupt transition. Also,
Okay.
these force curves are more like high dimensional objects because it's not just one number, but there's each value on the as a function of distance. So this makes us a little bit difficult to interpret, but that's a very good question.
Okay, thanks.
Yeah. Okay, so here's our experimental video on the left and the corresponding topological landscape on the right. Oh, and as we can see in these videos, what we eventually find is that the landscapes have two peaks. And as before, by marking the most dominant 14 motifs from the experiment, we can find that each peak corresponds to the most dominant motifs, and the first peak was indeed M1, and the second peak was indeed M2. Now, over time, the step-organized structure transitions to a more crystal-like structure, and this is also reflected in the landscapes as M1 peaks grow stronger over time. And as before, we can see this even more quantitatively if we focus on this M1, M2 axis, slicing our landscape through it. Here we see how this topological landscape sliced along M1, M2 axis change over time. Initially, with the purple curves, we see that M2 is the most likely configuration, but as time goes, there is a time point where M1 and M2 are equally likely, and later, as systems keep going, M1 becomes more likely than M2. Again, we find that our system is transitioning from M2 dominated state to M1 dominated state. And also, this is again through a coexistent state. So, combined with our model result, this topological landscape provides a simple explanation of how our living matter transitioned its self-organized structure over time. And now I'm moving on, and if we do not have any questions. So what we now have is a time series of topological landscapes. At this point, we realize an interesting parallelism that leads to the cherry on top of this talk. For our living matter, by assigning coordinates for each topological motif, we arrived at a simple visual description that helped us better understand the system. Similarly, if we assign coordinates for each topological landscape, each curve on the left we may get another set of new insights on the self-organization dynamics. And to assign coordinates to landscape, we focused on the fact that each landscape is essentially a probability distribution, and between probability distribution, there is a concept called callback linear divergence or KL divergence, which quantifies the dissimilarity between these landscape probability distributions or our topological landscapes. Basically, it is a measure of how one distribution diverges from a second reference distribution, so that if the distribution is far from the reference, then it will have high KL divergence, and if it's close to reference, then it will have low KL divergence. And this is how we use this KL divergence to construct our topological flowscape using M1 and M2 as reference point. On x-axis, we have the KL divergence from a M2 dominated distribution, which quantifies the dissimilarity from M2 essentially. On y-axis, we have KL divergence from M1 dominated distribution, which will tell the dissimilarity from M1 of our probability landscape, our topological landscape. And as you may expect, the system is initially far away from M1, and in its final state will be close to M1. The question is, what happens in between? Like how the landscape reveals the two motifs M1 and M2, this flowscape shown here, mapping each topological landscape over time to this KL divergence space, reveals two modes of dynamics in our self-organization. In early time, basically the system runs toward both M1 and M2 as going toward this center. However, and in late time, the system is still approaching toward M1, but is turning away from M2. And as we saw in the velocity polarization, this transition is clearly abrupt and just two-step instead of being more like continuous. Beyond the directions, we can also ask what happens to the speed of the transition. And this can be done by choosing. One way to think about this is to choose a different set of references. For example, the initial landscape and the final landscape, so that the x-axis here tells the dissimilarity from the initial point, and the y-axis shows the dissimilarity from the final point. And basically the diagonal line would tell us the progress of this self-organization from initial to a final form. Here, we are showing such progress from initial to final landscape. In other words, the dissimilarity from initial minus the dissimilarity from final. And intuitively, this is basically like a progress bar that tells how much the self-organization process has gone through over time. And what we immediately notice is initially coinciding with the traveling state in terms of the velocity polarization, the system is transitioning pretty fast in terms of its topological configuration, while in later, the system still transitions, but the transition has gotten much slower. In the end, what this flowscape does for us is that it reveals that there are two states in our experimentally observed self-organization process. In early time, the self-organization process is characterized with a fast topological turnover toward both M1 and M2. For late times, the self-organization process is via a slow topological turnover toward M1 and away from M2. And as we recall where we have started from, we feel that we have walked through a quite long road toward a better intuitions for our self-organizing living matter. And I'll show the summary of our finding with laboratory experiment, interaction inference, and numerical simulations, we observed and identified a diverse set of self-organized states of non-reciprocal matter. With topological landscapes, we found out that the non-reciprocity drives all structural transitions between M1, the perfect crystal, and M2, the almost perfect crystal with 5 defect. In addition, the way that the system transitions is through a coexistent state, and this has led to more powerful intuitions and additional testable hypotheses for our system. Finally, with topological flowscape, we find that there are not only these two states in terms of the velocity polarization, but also two states in terms of the self-organization process, how the system is approaching from initial state toward the final state. And now, while that was the takeaway for specifically for our question, like non-reciprocity, our system, we feel that these tools we developed, the topological landscape and flowscape, has even bigger possibilities. First, this converting from the living matter into a topological flowscape basically allowed us to reveal this emergent order, in our case, the M1, M2 axis, for any kind of the complex structures. And this, in principle, could be applied to other systems as well. Finally, also, mapping from topological landscape to a flowscape was essentially for us to map the self-organization process as an informational process, which was only possible after we constructed this topological landscapes, which is the probability distribution in terms of the representation of this self-organized structures. And we hope that these lessons we earned and kind of system-specific intuitions that we got from the process could also convince you that this is a method that is worth thinking about. With this, I'd like to end my talk. Thank you all for listening and also for nice questions.
Okay, any questions? You can just unmute yourself. Yeah.
What about 7 disclinations, 7 coordinated particles?
Excellent point. Yes. Indeed, what we do find in our system is that the 7 defects are more difficult to find than the 5 defects. My naive intuition is that if we look into how we construct the landscape, basically it was not that the system was already kind of close to the crystal, and okay, maybe this is a good picture. We do find some motifs with crystal. For example, this particular motif M4, the center one on the top is the one with basically a 5 to 7 defect pair. And it's just that the 2 defects, the 5 and 7 defects, are not equally represented in the system. And my sense is that for other systems, if we do the same approach, maybe we might find something different. But yes.
So for systems that fill space, which I know this one doesn't, you would expect 5s and 7s to be roughly in balance depending on the
Yes.
overall topology. So is there something about the active dynamics that encourages there to be clusters with a net positive disclination charge?
I think it's rather, yes, I do not have a good slide here, but basically what we also did was to go back to the experimental video and label M1 and M2s that we see on the, let's say, physical space. And what we generally find is that M1 corresponds to the bulk of these small crystals, while M2 corresponds to the boundary of the crystals. And in that sense, I think the asymmetry between M1 and M2 comes from the fact that this is kind of open system with large gaps, and especially those gaps are promoting the 5 defects, not 7 defects, when we just recast them into the network topology. And I think it's an interesting question. Then what is the other way or like other kind of type of systems that would maybe like even show more 7 defects than 5 defects perhaps. But this is an open question.
Okay, just quick clarification based
Yes.
on what Dan asked and your response to that. Does it matter that the meniscus is curved?
Mm.
Because if the meniscus is sufficiently curved, then it's not a plane, and then you could imagine that some of the excess of plus defects is because of that.
That's an excellent point. I think that indeed in our lab, while we did some experiments with controlled meniscus, we didn't explore the regime of even stronger meniscus effect than what we see in these kind of default scenarios. And it'll be good to revisit what's happening. Maybe one to think of another experiment that could be interesting in this aspect was there was recent attempts to do some experiments with these starfish embryos on a cone where there is a defect in the center, and that could be maybe a good platform to explore this idea of what may happen. But that's like some point things.
The cone is perhaps an extreme example, but you could certainly have some kind of post in the middle with a curtain of meniscus around it that would
Mhm.
allow you to probe different kinds of
Ah,
curvature.
that's also true. Yes.
I have a question on your modeling plot of the log of probability versus M1, M2.
Yes.
When I see that curves, yeah, I see that it seems that there is a non-monotonic behavior when you going up from n to
Yes.
0 to 0.75 and then to
Mhm.
1.25. But it seems that in the experiment, you just gradually downwards. So you do not see
Yes.
that non-monotonic behavior. So is there any reason for that?
That's an excellent point, and I will tell you what we understand. First, in terms of model, what happens with this non-monotonic behavior in terms of non-reciprocity actually seems real. And what we also find is that similar to what we have in this let's say visualization, we could also basically define what we call the hex, or basically a topological distance from a perfectly crystalline structure of each structure. And that also shows that around, let's say, non-reciprocity of 0.5, the system is actually closer to the crystal than what you get from a reciprocal case. And in short, this is probably because even the reciprocal case, we still have not a, let's say, monodispersed cluster. We have a bidispersed cluster. And this non-reciprocity, small amount of non-reciprocity is actually doing almost an annealing of the system so that it disrupts these more, let's say, unstable structures and selects more like stable kind of crystalline structures. So that is what we understand in terms of the model. And now, as you definitely, you are definitely right that the trajectory in the flowscape in terms of the experiment does not seem to capture the same trajectory. And in fact, we can actually look into this by side trajectories. On the right, we have that experimental landscape, flowscape, but on the right, what we have is the flowscape for, let's say, simulations, where each point corresponds to each collection of, let's say, the snapshots from the simulation with fixed non-reciprocity. And this also kind of confirms that the two trajectories, one based on the change in non-reciprocity and one based on the change over time, are different. We have some thoughts on why these two can be different. And in general, we think that since this coexistent state in terms of analogy could also imply existence of hysteresis, that kind of initial state difference dependence could also lead to more different behaviors. But And I have more to say about like this, but maybe this is too long answer already. But is that
Thank you.
okay?
Okay, so I will stop the recording now, and then after that,